So good morning, I'm Augusto, and today I'm gonna talk about the paper Understanding Cancer Mechanisms Through Network Dynamics. In this paper, the authors, they provide a review and a general picture of the mathematical methods that can be used to tackle and to study cancer mechanisms, and the framework that they use is the network theory. So as an introduction, with the recent advances in with technology and computational method, there has been a huge increase in the amount of information available in the biology side. So we have a huge number of genes, proteins, and interactions inside the organism, and with this huge amount of data, we need a different approach to better study and better understand how all how all of these elements interact and the functions that they play inside an organism. So one of the mathematical methods that can be used to study this is using network theory, in which you can build the networks with the elements and their interactions. And when we go to the cancer side, we can have simple networks, such like the first one, when we have a signaling pathway in which the cells are able to turn on or switch off some specific functions in the cell by receiving external stimuli or other kind of stimuli by activating or deactivating some functions and other proteins. We can go adding some complexity into this network by adding crosstalks and feedback loops. So we have a more complex signaling network. And in another level, we can, if we want to see the whole level of protein or the whole level of genes in a network, we can have a large network with all the genes or proteins in an organism and all their interactions. And going to a deeper level, we have to consider all the environmental effects of the cell and organism inside the specific tumor cells. So we have a more complex dynamics in the network. And to study these networks, there is two approaches. We can consider static networks, or we can go to the dynamical networks. In the static analysis, one of the most used networks is the protein-protein interaction networks, in which we have the map of all the proteins of an organism and all the interactions between these proteins. So we can see the organization of these proteins and also some of the functions. And this static analysis provide the qualitative results, and we can use this to predict new disease genes or to further study the ones that you already know. We can also identify potential cancer drug targets or also identify new cancer biomarkers that are proteins or set of proteins that have specific functions that characterize the presence of a cancer cell or something like that. So the static analysis can provide us with some good results to begin the study of a cancer network or a cancer system. But if you want to go deep and consider more aspects of the network, we have to include the dynamics inside the networks. And by using dynamics, we have some non-static networks, and we can map the flow of reactions and also include the perturbations caused by oncogenes or cancer drugs inside this network. So dynamics can make it, we can use dynamic to study these perturbations in the networks. And another important aspect when we consider dynamics is that we can link the structural properties of the network with dynamics by using what is called the network motifs that are sub-networks inside a network. And these sub-networks or these motifs, they have specific functions inside the network or inside the organism. And the two approaches to include dynamics in the analysis are by going through Boolean networks or using differential equations. The first one is the Boolean networks, when the gene expression is quantized as two states, on or off. So you have some pathway networks with the genes or the proteins that are transcribed by the genes, and they can be in one of the two states, on or off. And the interactions of these genes can be responsible for activating another one or inhibiting the function of another one. And then you have a Boolean network. And the analysis of the network is made by Boolean algebra, which is using the logical operators like and, or, and not, or the variations of these operators. And one of the greatest advantages of the Boolean networks is that we can handle large networks. So we can have hundreds or maybe thousands of genes in the simulations. And here's an example that is cited in the paper, which we have a pathway network with all these, with 27 different elements, and we have the green lines are the genes or the proteins that activate the other ones, and the red ones are the inhibition relations. So when you simulate these networks, the Boolean networks, they don't consider the aspect of time. The simulations occur simultaneously in the same step. And in the end of the simulation, the result is what's called a stable state graph, when you have the combination of all of the elements involved and all of the possible states that they can have. So they can be, the empty dots are when they are on, they're off, and the black dots are when they are on. So we have the combination of the possible states of these elements that will represent the phenotypes of this pathway. So we can, this is a qualitative analysis of the network, but what is good is that you don't need to consider any parameter of the organism, parameter of the network, like forgot. But you can do this analysis. We have a good approximation of the signaling and the regulatory network without considering
2 minutes.
Okay, without having to take into account any parameter of the organism or the network. But if you want to go to another level of complexity and have a better description of the network and having quantitative results, you can use networks of ordinary differential equations, in which you have quantitative descriptions, but you have to consider parameters of the network, rate constants, and all of these aspects to solve and to get the results from the differential equations. And one of the applications is that you can combine some pathway information with the structure of the proteins to have the informations about the effects of mutations in proteins. So combining the ordinary differential equations for the pathway with the structure of specific proteins, you have the differential equations, and you can analyze the effects of the mutations into this protein. Another application of differential equation is you can model the interactions between the cancer cells and the surroundings inside the cell environment using, for example, fluid dynamics through the cellular medium and external medium. And you can also model the response of tumor cells to therapeutic treatments to see the effect of some drugs, if they are effective or not. in the treatment of specific types of cancer. So as a conclusion, the static networks, they provide an informative result, especially at the beginning of the study, so we can identify potential targets for the further studies. And the Boolean networks, they can tackle and they can consider hundreds of genes, but they only provide a qualitative approach. But these qualitative results are good because they provide a good approximation of how this signaling and how this regulatory network works in the organism. And in the end, ordinary differential equations we can represent protein concentrations and also have a more sophisticated and quantitative approach when studying the effects of cancer and cancer drugs. So combining these methods, you can have a pretty good understanding of how cancer works and how we can develop some drugs and approaches to treat and maybe cure someone. So this is it for my part.
Okay, thank you. Questions? Perguntas?